As long as we don't get eaten by the tornado, we should be able to get through this. So my name is Karen Sachs, and I wear a few hats. So I have a consulting company called Next Generation Analytics. I'm also working with the lab at Northeastern. And I also have, along with Eric Mockler, who did most of the work that I'm presenting today, we have a longevity company where we are developing a molecular model that's personalized to individual people. Now, in longevity, it's very, very difficult to say, is this perturbation the correct perturbation? But in cancer setting, we have a little bit more ground truth to work with. And also, I've done also a fair bit of work on cancer in my career. And so in collaboration with Ryan Lee and Zohar Sachs, who are both at UMN, Zohar Sachs is an AML expert, and my sister, and Ben Gyori, who's at Northeastern, who will come up later in the slides, we said, let's try our methods and let's try other methods that we have seen out in the literature in AML. So this is a slice of that, what you guys will see today. So just 2 bio slides. Cells respond to their environment through a monstrously complex control circuit. I think people in this room already know that, but I'm just very quickly. Cells respond to signaling responses in the body, and they respond by making a decision, a fate decision, and how they're going to act, what they're going to do. And inside the cell is a molecular network, this monstrously complex control circuit. And that control circuit can break down in disease. So for example, if the control circuit decides to proliferate and migrate when it shouldn't, those become the hallmarks of cancer, and the person gets very sick. And so we would like to understand these circuits, and we would also like to know, can we perturb them in such a manner that we get the system back to a wild type state and therefore cure or treat the disease? So that's the setting in which we're working. And the question that I'm addressing in today's short talk is, well, a slice of this, can AI models predict the perturbations in genetic regulatory networks? It's not only genetic regulatory networks, but today I'm kind of focusing on genetic. It's also the upstream signaling networks that we care about. But today we're kind of looking more at genetic regulatory networks. So just very quickly, when we are, as systems biologists, when we are trying to model these complex circuits, we might start with something like clustering, right? You guys have all, this pointer is not working. You guys have probably seen, so you guys have probably seen, clustering results when I have a data set. Oh, thank you so much. Okay, so if I have a data set and I want to get some intuition on it, I might cluster and I might see some kind of gene list analysis where I say, okay, in the AML samples, I see that PI3 kinase or RAS pathway is more active, or I might say this particular patient has more RAS and this other patient has more whatever, AKT, I don't know, right? So we can characterize using this very broad kind of gene list or clustering approach. We can also go all the way to the other extreme and make these detailed dynamical systems models that really show you the whole pathway and how it works. Now, these are beautiful, amazing models, but they're kind of impossible to do at scale. So if you're trying to capture as much as you can about this complex biological circuitry, you might end up at this sweet spot here in the middle where you're looking at causal models. And causal models are trying to capture the causal circuitry more than anything else. So they're saying, can I capture as much as I can of the detail without going to so much detail that's prohibitive in terms of modeling? So it's this sweet spot of trying to capture causality to an extent. Now, they have shown great progress in a lot of different domains, including biology, but which I don't have time to talk about, but see me after if you're interested. But there are concerns regarding performance. There's a lot of concerns regarding performance. Here are 2 papers that are attempted to benchmark these causal methods. And basically these papers have said, these methods don't work very well at all in the context of genetic regulatory networks. Now, I can tell you I'm an expert in this. I've been working in this field for 20 years, possibly longer than most people on the planet. And I can tell you that part of the problem is the way they're benchmarking and the way that they're assessing how well these methods are working. And part of the problem is algorithmic, and hopefully we'll get back to it at the end of the talk if I have enough time. I think it's actually a really exciting and really interesting domain of study. Part of it is algorithmic and so on. So hopefully we'll get back to that, and if not, come talk to me. Oh, I'm sorry, that was supposed to be a build. Okay. Oh, no, sorry. Okay. So apologies. So the benchmarking studies have basically gone through and said, do you fit the information that I see in Reactome, which is one of the databases that's supposed to capture canonical signaling and canonical networks? Do you represent that well? And they also do some statistical metrics. And like I said, I think those are limited to an extent. Nevertheless, they haven't shown remarkable performance. Nobody is impressed with the performance they've shown so far. So enter deep learning. So deep learning has been around now for a while. Some of the performance in deep learning is mind-blowing. All of us have used LLMs. They're really remarkably powerful. So people have said, well, concomitantly with deep learning improvements in performance, we have also seen a proliferation of biological measurement approaches that will let us perturb and measure the components of these circuits, of the genetic regulatory networks. Perturb-seq is probably the most famous, right? And in Perturb-seq, you can do genomic scale perturbations where you turn off individual genes and then measure many cells for each perturbation, where you can see what happened to the system after I turn off a gene. Now, it doesn't work for literally every gene, but it's quite genomic in scale. And so people said, like this paper, sorry, the citation is cut off here. It's Lutfa Lahi et al., Molecular Systems Bio 2023. People said, well, can we take this big perturbational data sets and can we implicitly learn the wiring of the biological circuit without even worrying about, RAF goes to MEK goes to ERK? We're just going to dump in a bunch of data, put it in a black box, and dump out a prediction for for some combination of perturbations, and therefore figure out how to cure an individual person's tumor, for example, or other therapeutic applications. This is pretty cool, but does it work well? I would say not yet. It might eventually work, which is pretty exciting. But right now, I would say it's not the model. The deep learning models really are powerful if you have enough data and enough computation. But the data is not there. So right now it requires exhaustive gene perturbations, which we actually can't get technically using Perturb-seq. And if we could, it would be very expensive, but we actually can't get it. And we also need to implicitly capture that biological circuitry. Now, if you think about it, that's really hard to do. Biological circuitry is condition dependent, it's cell type dependent, it's organism dependent. And maybe you can argue that you fuse transfer learning or something like that. You can capitalize on limited data. But at the end of the day, this is my personal opinion, the reason it's not working quite yet is because of that. I would also argue, and maybe this is my bias as a causal AI person, but I would argue that causality matters. And it matters partly for insights. So as biologists, we care how the system works. And it also matters partly for generalizing. So if I now have a model, a black box model, and I predict the perturbation for this patient, and now it doesn't work for this other AML patient, now what do I do with that? Right? I have no idea. The black box didn't work and I don't know why. But if I have a causal model, I can go in and say, is there a mutation that explains a rewiring event that explains how mechanistically this patient didn't respond to the drug? So I can do much more. I can go much farther. There is a middle ground that I want to talk about today, which I was really excited by. There was a paper by Yusuf Rouhani in 2023, made this beautiful Nature Biotech paper where he said, you know what, we're going to start with the perturbations. We're going to take unperturbed data. We're going to take perturbations, and we're going to try to capture the causality via embeddings. And the embeddings were from some literature prior knowledge networks and also from gene co-expression. We're going to try to capture the causality in the embeddings, and then we're going to put them into kind of a black box model. So it's this nice sweet spot. I think it was a brilliant piece of work. I loved it. And so this is essentially what they did. Oh no, I'm sorry, our build didn't work. Okay, so I don't have the gears model up here, but the gears model that I just explained, I guess, is essentially what it did, so that's okay. And I wouldn't say the performance was mind-blowing. I would say the idea was beautiful, and the performance was compelling, but not mind-blowing. And part of the reason, like I alluded to before, is that they're training on 2 different types of cells. We have many more than 2 different types of cells, and also the ability to combine perturbations when you've captured the circuitry to the best of your ability, but using kind of not the most powerful prior knowledge networks and so on, is going to stay fairly limited. There's another model that I wanted to, oh, I see, I don't have the, Oh, I'm screwed up. I'm sorry, I screwed up. I put the same diagram for both. There was another model that came after gears. So there were a number of efforts that came after gears that really literally use some of the gears code. So they're very much like a complement to Rouhani, I would say. It was very much building directly on what gears made. And here's an example of one. There's a BioDSNN that is by Tan et al. Briefings in Bioinformatics 2025. Sorry that you can't see that. And they, very, very, very broadly, they improved it in two different ways. One, by capturing the expression data itself in a bit of a more robust way that accounts for the noise in the data a bit better. And two, by using what's called an attention mechanism, which many of you are probably familiar with, and masking the attention mechanism in such a way that they're hoping to capture the biological, the known biological pathway in a better, more effective way. So when you're trying to predict the effect of a perturbation, you're taking into account the context of the reaction of the pathway that it's known to be in, and you're trying to use masking to capture which genes are relevant for this particular prediction, which genes are not relevant. So that's very high level. You can read the paper too, but high level, we said, let's look at two different models and let's get an idea of how similar they are or how different they are from each other. And so in our context, we said, okay, let's use it in the context of AML. There's a beautiful paper from Van Galen et al. In Cell of 2019 that you can't see at the bottom here. And what they did was take a bunch of patients and some controls, and they took bone marrow and they measured, so AML is a cancer that's in the bone marrow, and they measured not only gene expression, single cell RNA-seq, but also the genetic profile of those people. And so we said, okay, can we use the predictive models, those two predictive models, to say, can I cure the disease? That is to say, does there exist a perturbation such that the malignant cell starts to look more like the cancer, the normal cell? And we can also ask in the other direction, is there a perturbation such that the normal starts to look like the malignant? Now, you might say, why would I care about this? Why would I want to induce cancer? And that's, of course, for the sake of identifying putative causal or driver mutations using this method. It was also, we had kind of two goals. One was to maybe learn something about AML and learn something about the methods, but also to kind of say, how different are these methods? So you use two different, we know that deep learning is an art more than a science, frankly. And so can we learn something about, how different these are? So the high level idea here is that we have this kind of high dimensional histogram that I'm depicting here in 3D, but it's this high dimensional thing with all the different genes. And I think they filtered it down to approximately 5,000 genes, all these different genes, and they represent the distribution that is unperturbed or that is perturbed. And after it's perturbed, you can get one prediction from gears and one prediction from BioDSNN. And you can kind of say, do I see predictions that seem to move the malignant towards the normal or the normal towards the malignant? So that's the gist. And we're going to represent these high dimensional histograms just by their centroid for the sake of simplicity right now. Okay, so just really quickly, let's compare these models. How am I on time? Okay, squeezed. So really quickly, we're going to just compare the models. They had what I would call reasonably high consistency. It was a little bit higher than I expected. So this is the Pearson correlation by cell type. And what you're seeing there is over lots of different perturbations, how well correlated were the two models that we are comparing. And they're spread, and there's some few outliers. There's a few crazy outliers here at the bottom. But overall, I would say the correlation is not bad. This is Pearson, and this is Spearman. So not bad. I don't know. You guys could argue that it's not that good, but, and these are broken down by cell type. You can see here. So in AML, some of the most interesting cell types are GMP, the general myeloid progenitor, and HSC, the hematopoietic stem cell, where the cancer must originate. And so those are kind of broken down by cell type. And you can see that there are actually differences. And you see one patient on the left and one patient on the right. So these are just two different patients. And there are differences, but overall, reasonably well correlated. And we dug in and we said, okay, let's look at some of the individual perturbations. And so These are the most correlated perturbations in this patient, and these are the least correlated perturbations in these patients, and each dot here is one gene. And do you consider these? I don't know. So in terms of interpretation, I'm not sure exactly, but you see like a reasonable correlation. So are they similar? Yes and no. Okay, I'm going to rush ahead. So we said, okay, can we assess these predictions? So really quickly in the last couple minutes, we did have the mutation profile, and we do have a literature prior knowledge. I'm working with Ben Giori at Northeastern, who has compiled the most comprehensive prior knowledge network. It's an absolutely amazing resource. It reads the literature every single day and updates it every single day. It is unbelievably comprehensive. And so we said, let's assess these predictions. So these are taking the patient and asking, does a perturbation move it closer to the wild type state? And these are the genes that we found. So the algorithm is predicting that turning, this is Gears is predicting that turning these off will bring you closer to the wild type state. And I can dig into Indra and I can say, do you agree? Well, Indra very much agrees. Indra very much agrees that this patient's causal mutation, as shown in the mutation profile that they take in the clinic and that they reported in this paper, does indeed affect these genes. And I went on to look at a few others. And the bottom line, there's quite a bit of evidence that it does. And then we said, well, what about in the other model? So the other model found other genes. The other genes also had support in the literature. So which one is best, I still don't know to tell you, and maybe I can dig in and do a more thorough analysis, but both of them give compelling, interesting answers. Okay, so rushing ahead, in terms of what's next, just a very quick overview of what's next. I'm a causal AI person, and I really believe in thoroughly understanding the circuitry using something called causal discovery, where we take the prior knowledge network and we actually use the data to learn the circuitry. And then we can go on and make that into a deep model. That's where I want to go next. The way I think to do better than what we're doing even right now is to use Indra to infer upstream regulators, because mRNA doesn't actually do anything, right? The mRNA is not a good indicator of who's activating who. And lowly expressed genes, which are important regulators, don't often show up very well in the mRNA. So that's one thing. And the other thing is that there's new insights regarding causal discovery that we have under review that people are welcome to talk to me about. I actually can't see what that says. Oh, okay, great. But that's with questions, right? Okay, so so I'll finish here so you guys have questions. I did say that I would say something about working in biology as a computational person and vice versa. I'm really in the middle. I'm really, really, really a bridge person. I highly recommend being a bridge person. It's amazing. The more you understand the biology, the more you're able to say, oh, wow, look what we can do, and find new horizons in terms of what types of applications we can bring to biology, and also what are the important questions. And really understanding the important questions requires understanding the biologist. I would recommend, whichever side you're on, work very closely with the other side and learn as much as you can intuitively. Don't have to understand exactly how a deep model works, but you have to understand what it does in order to use it effectively. That's it. I have, I think, 2 minutes for questions.
Yes, that was wonderful. Can you say a little bit more about this connection between, say, dynamic system and, say, a deep learning? Because as I was listening to you, you really speak the language of dynamical systems, right? So looking for these perturbations that you cure or they get into cancer. So when could I learn more about this?
Yeah, so it's such a cool topic. So my PhD lab was an ODE modeling lab. Doug Lossenberger, if you know him at MIT, is an ODE guy. So I was kind of bathed in the soup of dynamical systems. I think they're amazing. So a dynamical systems model, for the audience, is a differential equation model that is supposed to tell you, I stimulate the EGFR pathway, and here is the entire dynamics of what happens, and that's really exciting. These models are not trying to capture dynamics. They are trying to capture the endpoint, which is steady state. Now, arguably in the clinic, what we care about is the new steady state.
But that's okay, same thing.
Yeah, it's not exactly the same thing, because you're right that you end up with the same thing, but the dynamical systems model is trying to show you the entirety of the dynamics. in the context of the prior knowledge network, it's very interesting because the prior knowledge network will report from the literature the transient interactions and the steady state interactions, but the model won't. And that's part of the insights that I alluded to in that previous slide. The model won't, so it's actually a really important conversation. But these models are trying to do a shortcut and to say, let's go directly to the endpoint, to steady state, and let's just implicitly learn the circuitry to the extent that we can. And as a causal AI person, I want to do both. I want to take the circuitry and also kind of use the machinery of deep learning and put them together. That's causal AI, right? But not actually trying to learn the dynamics. Even though these dynamics models are really exciting and interesting, and maybe they give you information that you can't get using these models that kind of cut to the chase directly and go to steady state.
So if I look at the web page, can I find somewhere to start from scratch learning and that's?
It depends on which question.
In general, this issue of the perturbation, perturbing the system going to cancer or going to cure.
Maybe chat with me afterwards,
Yeah.
because there's a whole body of literature.
Yeah.
Yeah. Or email me. My email's on the slide.
Sure.
Is there any other question for our speaker?